Hi, this is Ms. Coleman, and today we're going to talk about the structure and function of DNA. Discovering the structure of DNA. The structure was discovered in 1953 by James Watson and Francis Crick. They used cardboard and wire to construct a model of DNA seen to the right. It's called a double helix, in which two strands are wound around each other. So if you look at the picture, you can see that there's strands on the side and they're kind of coiled or twisted. And then there's little, kind of like, looks like steps or rungs of a ladder in the center, and those get twisted as well. DNA stands for deoxyribonucleic acid. So there's a D for deoxyribo, there's the N for nucleic, and then acid at the end. It's found only in the nucleus and contained in chromosomes. Remember from our study of the cell that the nucleus is the brain of the cell, so it makes sense that your information for heredity is contained in the brain of the cell. It's very important information. Chromosomes are composed of two different substances. The first is protein or histones, and DNA, which is your genetic material. So if you look at the picture to the right, you have your chromosomes, and they're tightly coiled and condensed. And then if you follow it over, here's your DNA double helix. So one strand, two strands, and here's the rungs of the ladder I was speaking of. The structure of DNA. DNA is an example of a polymer called a nucleic acid. From our study of biochemistry, we talked about monomers and polymers. So remember, poly means many. So the polymer is referring to the whole DNA molecule, and it's made up of little pieces or monomers, and those would be the nucleic acids. It is made up of smaller repeating subunits called nucleotides. Nucleotides we'll talk about later about what they consist of, but they make up the sides of the ladder and then part of the rungs in the middle. Nucleotides consist of a phosphate. The phosphate is represented right here in the circle. A sugar, which is deoxyribose. It's a pentagon, so 1, 2, 3, 4, 5 sides of the sugar. And the easiest way to identify sugars is by looking at the suffix at the end. Sugars end in OSE. So for example, glucose, pentose, things like that. They all end in OSE, so you can easily identify them as sugars. Nitrogen bases. These are represented by the rectangle right here, and they make up the center of the DNA molecule. DNA, a spiral ladder. The phosphate group of one nucleotide is linked to the sugar group of the next nucleotide. Those are the sides of the ladder, the part you would hang on to. Nitrogen bases are attached to the sugar of the nucleotide. Bases are attached to each other in the middle, the rungs of the ladder, by hydrogen bonds. Nitrogen bases. A nitrogen base is an organic ring structure that contains one or more atoms of nitrogen. So that's why it's called a nitrogen base. In DNA, there are 4 possible nitrogen bases. The first is called adenine, the second guanine, third cytosine, fourth thymine. When looking at nitrogen bases, what makes them easily identifiable is by also looking at the suffix at the end. So if you see a word and it ends in INE, it should pop into your head, hey, this is a nitrogen base. In this picture, you can see the sugar, phosphate, nitrogen base. Sugar, phosphate, nitrogen base. The nitrogen bases in the center, they kind of fit with each other like puzzle pieces. So G and C, G, C, C to G, T to A. Base pairing rules. So like I just mentioned, you notice a pattern in that picture. Only certain bases pair with one another. The bases in a strand are always going to pair in a particular way. So for example, adenine always pairs with thymine. Cytosine always pairs with guanine, and this can go backwards as well. So thymine would pair with adenine, guanine would pair with cytosine. A and T are connected by 2 hydrogen bonds. whereas C and G are connected by 3 hydrogen bonds. Oops. Next, on the double helix, the nucleotides combine to form 2 long chains, producing 1 long molecule. The 2 chains of nucleotides are joined by hydrogen bonds between the bases. This causes the molecule to twist and form a structure that resembles a coiled spring, and that's why we call it a double helix. So a little clarification on why it's called that. In the picture, you can see the sides of the strands are composed of the sugar and the phosphate. So this would all be sugars and phosphates on both sides. And in the center, you have your nitrogen bases. So A and C are paired together with 2 hydrogen bonds. C and G are paired together with 3 hydrogen bonds. Similarities in DNA. The genetic material in all living things is made of DNA. So it's what we have in common among all organisms. DNA is made up of the exact 4 bases no matter what kind of organism it is. The thing that makes something become a squirrel rather than a rosebush, or a person rather than a rosebush or a squirrel, has to do with the order of the nitrogen bases in the strand of DNA. This is very important for you to remember. You need to know that the order of the letters, so adenine, guanine, so the A, G, C, and T, the order that those are in is what makes us different. The more alike 2 organisms are, on the other hand, the more alike their order of nucleotides will be. So, for example, rodents and squirrels would have nucleotides in a specific order that's more alike because they're more related than a squirrel is to a rosebush, for example. Replication. Replication is when cells divide and there must be some kind of mechanism in place where it ensures an exact copy. So the new cell needs to get an exact copy of the DNA as the previous cell. Replication means to copy. So for example, say there's some kind of famous painting and you're in art class and you go and copy that famous painting. You're making a replica of the famous painting. This mechanism that the cell uses is called DNA replication. So you're making a copy of DNA. Each organism on Earth today has a nucleotide sequence that it received from its parents. So you received one from your parents, the squirrel received one from his parents, and so on. Steps. There are certain steps that a cell follows to replicate the DNA. The first is the DNA molecule unwinds. So it's tightly coiled and it starts to uncoil. The DNA molecule then unzips. So it's coming apart exactly like a zipper comes apart when you unzip it. An enzyme called DNA polymerase. Another interesting or important way for you to remember enzymes or identify them is that they end in ASE. So if you look at the suffix of this and you see a word that ends in ASE, you should think, hey, that's an enzyme. It breaks the hydrogen bonds between the nitrogen bases so the bases are exposed. So basically what that means is that they're coming apart. The third step is the exposed bases serve as a template. Next, the nucleotides line up. The phosphate group of one nucleotide bonds with the sugar of the next to create the new strands. Replication results in 2 molecules of DNA where there had only been one previously. The 2 molecules should be exact copies of each other. Each of the new identical strands of DNA has one of the original strands. Later in the unit, we'll talk about what happens when you don't get an exact copy, and that's when mutations occur. In this picture, there's a summary of DNA replication. So in the picture, you can see how it's uncoiled and the sides of the ladder have unzipped. So it looks almost exactly like a zipper right in this picture. Then it's going to be start to copy on both sides. There's copying on this side, and then in the opposite direction, it'll start to copy on this side. So you end up with 2 new strands. Lastly, what you're going to do is you're going to watch this short animation of DNA replication and then answer the questions that I gave to you in class today. Tomorrow, you're going to bring those questions with you, and those are going to serve as your entry ticket into class. Once everyone's turned in their entry tickets, I will go over the questions to make sure everyone has a good understanding of DNA structure, function, and replication, and then we'll get started on the day's activity. Thanks for watching and have a good evening. See y'all tomorrow.